Hello, my name is Jamia Marcy Yu, and today's date is May 8, 2022. I'll be presenting about chemoreception, the senses of smell and taste. So, what's chemoreception? It's the process by which organisms respond to chemical stimuli in their surroundings, typically through their taste and smell senses. Chemoreception is based on chemicals that operate as signals to regulate cell activity without being absorbed into the cell for metabolic function. So let's talk about taste and smell. Taste is also known as gustation, and smell is commonly known as olfaction. They are the most interlinked senses since both are involved in molecules of the stimulus entering the body and connecting to receptors. Our sense of smell allow us to detect the presence of food or other animals, whether it be prospective mates, predators, or prey, as well as other compounds in the environment that might affect our survival. Similarly, the sense of taste enables animals to distinguish between different sorts of meals. While the usefulness of one sense of smell is evident, what about one sense of taste? So, different types of meals can have different properties, which can be both either beneficial or detrimental to the animal. So, let's talk about taste and odor. So taste and odor stimuli are both molecules which are absorbed from the environment. Sweet, sour, bitter, salty, and umami are the major flavors recognized by us humans. So the first four flavors are, the basic ones, very self-explanatory. And then the flavor of umami, which is also known as savoriness, is caused by the amino acid L-glutamate. Furthermore, monosodium glutamate or MSG is frequently used in cooking to improve the delicious flavor of some meals. I myself use MSG a lot when I cook. Alright, so let's talk about the smell and taste in the brain. So, smell, it in the brain, the olfactory epithelium in humans is a collection of specialized olfactory receptors that spans around 5 centimeters square in the back of the nasal cavity. Each cilia of an olfactory sensory neuron has just one kind of receptor. And because the receptors are specialized to detect certain odorants, the bipolar neurons are likewise specialized. Here is a figure of a bipolar neuron. Let's go back. Okay, so when an odorant binds to the receptor or to a receptor that recognizes it, the sensory neuron then associated associated with the receptor is then activated. And through olfactory signals during that whole like smelling session, the olfactory signals are sent directly to the amygdala, followed by the hypothalamus, thalamus, and frontal cortex. So the last structure to which olfactory signals are directed is a cortical location in the temporal lobe structure. Memory, emotions, reproduction, and thinking are all processed by odor-sensitive regions of the brain. So, like, if you ever smelled like a particular candle, it could like bring you back to like a childhood memory. Like some people experience that, and it's very common. That's why, like, it says here, it relates to memory and emotions. Okay, then let's move on to taste. The primary taste organ is a taste bud, and through taste neurons in the tongue, esophagus, and the palate convey signals to the medulla in the brainstem. Taste signals are sent from the medulla to the thalamus and then to the major gustatory cortex. The medulla, thalamus, and cortex are responsible for separating information from different areas of the tongue. And as I mentioned before, the primary taste organ is the taste bud. A taste bud is a group of gustatory receptors, also known as taste cells, which are located in the papillae or the bumps on the tongue. And every 10 to 14 days, taste buds, taste cells are replaced. So that's cool. Molecules, also known as tastins, are absorbed in saliva where they bind to and stimulate the microvilli receptors. Okay. So odorants, they are odor molecules. They enter the nose and dissolve in the olfactory epithelium, the mucosa at the back of the nasal activity, as you can see on this figure. The olfactory epithelium is a collection of specialized olfactory receptors in the back of the nasal cavity that spans an area about 5 centimeters squared in humans, as I mentioned before. And an olfactory receptor, which is the dendrite of a specialized neuron, responds when it binds to certain molecules inhaled from environment by sending impulses and directly to the olfactory bulb of the brain. And just for fun fact, humans have about 12 million olfactory receptors, which are distributed among hundreds of different receptor types that respond to different odors. Okay, let's talk about pheromones. So, pheromone signals are sent not to the main olfactory bulb, but to a different neural structure that projects directly to the amygdala. We covered in this class that the amygdala is a brain center important in emotional reactions such as fear. The pheromonal signal then continues to the areas of the hypothalamus that are important and vital to the reproductive physiology and behavior. So, as you can see, here is the figure of what's the opposite of pheromones? Odors, stenches, stinks. Okay, so in summary, humans have 5 primary tastes: sweet, sour, bitter, salty, and umami. Each flavor has a distinct receptor type that reacts only to that flavor. Tastins enter the body and are dissolved in saliva. Taste cells are found in taste buds, which are found on 3 of the 4 types of papillae in the mouth. There are thousands of odorants, but only around 10,000 of them can be detected by humans. Like taste receptors, olfactory receptors respond to only one odorant. Odorants dissolve in the nasal mucosa, stimulating the olfactory sensory cells, and when these cells detect an odorant, they send signals to the main olfactory bulb and then to other areas of the brain, including the olfactory cortex. And here are my references. And thank you for listening to my presentation about chemoreception and the senses of smell and taste. I hope this presentation helped in any way, and I hope it was easy to follow and understand. Thank you.